Hi, I'm Tiffany, a member of the Neuroplicity team. Today we're going to talk about genes and behavior. Did you know that scientists can change behavior by changing DNA? Let's take a look at some research at Duke University that does exactly this. To gain more insight, we met with Dr. Scott Soderling to talk about his research on the actin cytoskeleton and how problems with related proteins may cause neurological disorders. Dr. Scott Soderling is an associate professor in the Department of Cell Biology and Neurobiology at Duke University.
The primary interest of my laboratory is the regulation of the actin cytoskeleton, which is a network of proteins that provide structure to cells. And in particular, we're interested in how this regulates the function and the development of the nervous system.
Dr. Soderling's lab has been studying how actin formation is regulated. The actin cytoskeleton is a network of proteins that help give structure to the cell. Actin is mainly located at the axon terminal of the neuron, where the cell sends signals, and in the dendrites of the neuron, where the cell receives signals. Actin can grow and shrink by adding or subtracting smaller units called G-actin. G-actin is the small circles I'm drawing here. In this way, actin can change the shape of neurons.
There was a key node in that pathway that directly activates actin formation. We call it polymerization in the cell at these sites of synaptic contact. And this protein complex is called ARP2/3 for actin-related proteins 2/3.
Actin-related protein 2/3, or ARP2/3, regulates actin polymerization, the process of binding several G-actin together. ARP2/3 does this by binding to actin so that it can make branches. This branching process is important because it allows the cell to reach out during neuronal development and make proper connections. Improper connections between neurons can lead to many neurological disorders, including intellectual disability, schizophrenia, and autism spectrum disorders. So maybe ARP2/3 plays a role. However, these disorders are very complex. They manifest through multiple behaviors, and many genes are thought to be involved.
One way of thinking about this is not, do these genes contribute to schizophrenia or autism, but maybe do these genes contribute to a common behavioral abnormality that's associated with both of these disorders?
To see if there was a connection between these abnormal behaviors and the ARP2/3 gene mutation, Dr. Soderling decided to create this gene mutation in mice. As a result, these mice showed behavioral abnormalities similar to schizophrenia. Who would have thought? Dr. Soderling's team was onto a great discovery. But how could they tell if a mouse had schizophrenia? Schizophrenia is a chronic human disorder that is characterized by hallucinations, disorganized speech, and delusions of grandeur.
Some of those things honestly, we just can't really test in mice. We can't ask the mice, did you have a hallucination? Do you think you're the king of all mice? We can't do those things.
However, they could quantify other behaviors like increased movement, also called hyperactivity, and stereotypy, which is a series of complex repeated movements. So his team asked if, after the gene mutation, did these mice show these behaviors and respond to antipsychotics just like humans with schizophrenia?
And it turned out that our mice had these abnormal behaviors, so they're hyperactive and they had stereotypy. And it turns out they respond to these antipsychotics as well.
Through these experiments, Dr. Soderling found that mutating actin-related genes led to schizophrenia-like behaviors in mice. But his story doesn't end there. Dr. Soderling also discovered a protein called warp. While ARP2/3 aids in actin polymerization, Dr. Soderling showed that warp helps to anchor the actin in the proper location in the neuron. At the same time Dr. Soderling was making this discovery, a research team in Germany identified a gene that caused intellectual disability in a young girl. Surprisingly, that gene makes warp, the same protein that Dr. Soderling had discovered.
That was very exciting for me because it was really the first suggestion, to me at least, that what I was working on probably fundamentally was critically important for how these neurons work in a way that's very relevant for diseases.
Dr. Soderling's main goal was to understand the regulation of actin cytoskeleton. However, through his research, he was able to make profound discoveries and contributions to the understanding of disorders like intellectual disability, schizophrenia, and autism spectrum disorders. Dr. Soderling's lab hopes to strengthen the understanding of actin pathways in neurological disorders through their future research. For more information on Dr. Soderling's research, visit his website, soderlinglab.cellbio.duke.edu.